Hello, history scholars, and welcome to this edition of Sharp Shorts, where we're going to talk about meiosis and sexual life cycles of the different organisms. And we'll start here. There's really two types of reproduction. There's asexual and sexual. And let me just get the full picture up here. This is, as we know, the sexual reproductive lifestyle of humans and really most animals. But what are the two modes? Well, asexual is just basically cloning of the parents. You see this mainly in of course, you see this in prokaryotes and protists, fungi. I'm a fungi. It can occur, and some plants it can occur. And asexual reproduction is it's mitosis. It's copying the genetic complement and producing, two cells, basically clones of each other. There's no genetic variability. One of the advantages of that, it all depends on the environment and things that are going on, but one of those is speed. It can happen rapidly. The young are very well adapted for the environment, and so can essentially go into adulthood right away. In small populations, like a small population of plants in an area, they can rapidly take over that area. When there's minimal plants and minimal amount of ability to do sexual reproduction because of the limited number of male and female gametes. So you get the idea. The disadvantage, of course, is that there's no genetic variability, which is key to the variety of things we have in life. And so if there was a catastrophe of some sort of environmental, you could wipe out a whole population. That's just some of the things that can occur. Sexual reproduction is fusion of gametes. Okay? And we talked about mitosis and cell division. Meiosis is another cell division, but what we have to ensure is that our gametes are haploid. Okay? Humans have 46 chromosomes, 23 from mom, 23 from dad. We're diploid organisms, okay? But as we develop those gametes, we need to make sure that they only have 23 of each chromosome, so when the gametes fuse through random fertilization, then you have a diploid zygote with the exact complement of DNA. So meiosis has to be different, and not just cloning a exact replica. In fact, you don't. You don't get exact replicas in your daughter cells. You get variability, which gives us are different and makes you special. But through that meiosis and fertilization, you have to make sure that you have that haploid. The big advantage, of course, is that variability, that ability to adapt to different environments, that ability. The disadvantages, of course, is you need a male and a female, or at least male and female gametes, to produce, which in small populations of plants or animals that are going extinct or things like that, is a problem, can be a problem. The disadvantages, of course, are the time. It takes time for gestation, and often those zygotes and those small organisms and babies and stuff are not ready for the environment, and they need to be nurtured much longer. And so, that gives you an idea that there's a place in the ecosystem for both, depending on, animals, of course, just do sexual reproduction, but for plants and other organisms, there are advantages and disadvantages to both. But the big genetic variability in life that allows for the large variety and the adaptability to different environments as organisms compete for food and space is through sexual reproduction and the fusion of those gametes. Okay. Now, we'll talk about with humans, the key thing in meiosis is to make sure that each gamete has a copy of each of, now here's the karyotype of a specific human. Karyotype is just a picture of, you have two number ones, two number twos, one from mom, one from dad, all the way through with your autosomes. And then you have sex cells. This is a male because there's an X and a Y, but you have those sex chromosomes. And the key is that each gamete has one of these. You have 46, your gametes have to be cut in half. Okay. What we're going to see in the key to this is that what we need is your homologous, both your ones, after they have been copied, to pair up and line up. And that's what we'll see in the steps of meiosis. Okay. Again, and this is what we mean by homologous pairs. Here you see karyotype for male, karyotype for female. This happens to be the males, but once this went through the synthesis phase of interphase, you can see here that you have homologous pairs. These were both the ones, they copy themselves. You can see they're held together at their centromeres, but these are what are called homologs. One of the keys to meiosis is that in that first phase of meiosis, that they pair up together to ensure that each gamete is going to get a copy of each chromosome 1 through 23. Okay? And this is this illustration. And don't get confused by the numbers. Here we have interphase, and they're going to copy. Say these are both chromosome number 1. These are homologous. You have 92. But this first phase of mitosis is to split these homologous pairs. And so that's called the reduction phase because you cut the number of chromosomes in half. Don't get confused by the fact that they're copied and how many sister chromatids they are. And then the second phase of meiosis is really just mitosis. It's just dividing this cell into two. It's just after the reduction phase, you're going to have 23 chromosomes in each, or the haploid number. But meiosis 1 is the reducing phase. Meiosis 2 is basically a mitosis phase. And what you get different than mitosis is 4 daughter cells that are distinctly different, as opposed to mitosis, where you get 2 cells that are genetically the same. Okay, so like mitosis, preceded by copying chromosomes, meiosis has 2 divisions rather than 1 to produce 4 daughter cells that are genetically different rather than 2 daughter cells that are the same. Okay, so again, meiosis 1 is a reducing, where you split the homologs that are paired up. Meiosis 2 is basically mitosis to equalize everything out, and you get 4 daughter cells. Here we go. So it's all preceded by interphase, which is the same. G1, the synthesis phase, G2, and here we're coming off of G2. Chromosomes are condensing. You head into prophase. You can see the chromosomes have condensed, the nuclear envelope is gone, the spindle apparatus is forming. What's different here is now the homologs have paired up, and this is the difference from mitosis. Where mitosis, the chromosomes can just copy themselves and line up any way they want in metaphase. Here you have to have the homologs pair up. So both the 1s, both the 2s, both the 3s pair up. One of the things that leads to that genetic variation then is what's called crossing over. And you can see that as these things have paired up next to each other, they have made a bond that's called a chiasmata. They've been hooked together, and what they're going to do, and you can see it by the color changes, is they're going to swap pieces of chromosome. And that's why those daughter cells are never genetically alike, and those offspring of any organism that goes through sexual reproduction is not exactly like their parents, even though they'll have characteristics. Other than that, the phases are the same. We've had the condensing, we have the tubule formation, we have different here now. We have the homologs lining up on the metaphase plate. We have the spindles attaching to the kinetochore. That again is one of those checks. And what you have in anaphase 1 is just separating the homologs, reducing the number to the haploid number. Of course, if for some reason one of those spindles didn't attach to one side of the kinetochore, you could have some non-disjunction, and that would then alter the appropriate number of genes heading out of meiosis 1. Here we have the end where you have telophase 1. You can see that you're getting the cleavage furrow. This is in an animal. In cytokinesis, the nuclear envelope is starting to reform. The spindle apparatus is separate, and again, broken down after that anaphase separation and going back to filling out the cytoskeleton. And then you go into meiosis 2, which is what they call the equational phase, or basically it's mitosis. Why not another round of interphase? I think that makes sense. We don't want to copy the DNA again. We want that haploid number so that when we fuse those zygotes, or fuse those gametes, that the zygote then has the appropriate amount of DNA. You can see here now that if nobody told you and you were looking at it under the microscope and you didn't know it was in meiosis, you didn't see anything going on here, it would look just like mitosis. You see in prophase 2, you have the spindle formation again, condensing of the chromosomes. Now, you just have the lining up of the metaphase plate of the sister chromatids, because this is the haploid number. You have attachment of the kinetochores to the spindle, and then again at anaphase you have the separation now of the sister chromatids. And again, if for some reason one of those spindles didn't attach to kinetochore, you could have non-disjunction, in which case you could get an inappropriate amount of DNA. That's something that can happen. Down syndrome. Down syndrome is a result of non-disjunction at chromosome 21 in humans. So what happened at some point was that maybe at anaphase 2, the sister chromatids didn't, so one of these daughter cells at cytokinesis and telophase, when I produced the 4, one of them would have 22 chromosomes, one of them would have essentially 24, because you'd have two 21s. And Down syndrome is called trisomy 21. You have, after fertilization, you have 3 chromosome 21s, which leads to a different production of proteins and the phenotypic characteristics you see of folks with Down syndrome. What I have here is a chart, and I'm not really going to run through it, but this is out of your book, and it's really good to see the comparison that they are very similar, but they have differences just to account for the fact that the gametes have to be haploid, and they have to have chromosomes 1 through 23. So you have to make sure in meiosis 1 that the homologs are paired up in prophase, and then separate those homologs, and then in meiosis 2 that you just separate like mitosis. Okay. Our next film, as we will go through here, is going to talk about the different sexual life cycles of different organisms. They all that do sexual reproduction all do have meiosis and fertilization, but it's how that goes in creating the organism. Until then, Scholar on Eastview.